how the fertilization is taking place in the flowering plants. This chapter we are going to discuss now. Already we discussed general characters of the embryology, introduction, and further sub embryology topics, and microsporogenesis, anther, anther structure, we have studied, anther tears, endothecium, middle layer, tapetum, stomium cells, and microsporangiums, and microsporogenesis, germination of the microspore, pollen structure, that all we have studied. And then in the second chapter we have studied ovule, gynoecium structure, ovary, in the ovary ovules, types of the ovule structure, ovule type, and megasporogenesis, embryo sac, how the embryo sac development is taking place, types of the embryo sac, and development of the embryo, development of the megaspore or embryo sac that we discussed. And then now we are going to discuss the fertilization process, how the fusion of male and female gamete is taking place. That we will discuss. The whole process, important process in the formation of seeds and next generation plants. This fertilization process is very much needed for the genetical exchange, gene transformation, and modification, and formation of new plants. Formation of embryo to seed, seed to plant. You know, the basic flower structure. You know the basic flower structure? That basic flower structure you can see. You can see the basic flower structure, how it is. I already told you. What are there? The pedicel and sepals, calyx, petals, corolla, and androecium, and at the middle center, gynoecium. Androecium, there is a filament and anther. Anther and filament in the stamen. The stamen structure, androecium structure, anther and filaments are there. In the gynoecium, ovary, style, apical part, terminal part, stigma. These three are there in the pistil. That means in the gynoecium. The fusion of these two, pollen from the anther, pollen reaching toward the stigma is called as pollination. That also we have discussed. Pollination. Pollination means reaching of the pollen toward the stigma. Either any one of the mode, either biotic factors or abiotic factors may be involved in the transformation of pollen toward the stigma. That is nothing but pollination. Either by biotic, that means either by insects or birds. Birds, that is ornithopily, insects, entomopily, if snails, chiropteripily, like that, myrmecopily, if the ants, and olcopily, that is by snakes, like that. Abiotic factors may be the air pollination, and aquatic water pollination. Whatever it is the way, the reaching of the pollen toward the stigma is needed for the fertilization. The first pioneer process, the premier first process in the fertilization is the pollination, reaching of the pollen toward the stigma. That is the first step, premier step in the fertilization. In the exam asks the question, write about the fertilization process in the flowering plant. Or write about the embryo sac which is ready to fertile. That is essay question. These two essay questions come from these. Okay, fertilization mechanism and embryo sac which is ready to fertile. That two essay questions will come raised from this chapter. Okay, that we will discuss here. You see, in the fertilization process, what's happening here in the fertilization? Fertilization means, what does it mean, the fertilization? I already told you. What does it mean, the fertilization? Anybody is there? Any student is there? Fertilization means fusion of male and female gamete. Fusion of male and female gamete. Female and male gamete. Fusion of male and female gamete is called as fertilization. Usually, in angiosperm, double fertilization will take place. That means fertilization will take 2 times. I already told you this fertilization process first identified, first reported by Strasburger. Who is that? Fertilization process first identified by Strasburger is the embryologist. Is the embryologist. Father of embryology is the Strasburger. You know, father of Indian embryologist? Do you know? P. Maheshwari. Panchanan Maheshwari is a gent, man, not woman. P. Maheshwari is the father of Indian embryology. Okay, you see, the double fertilization will take place. That the male gamete fusing with the egg cell is called as syngamy. That is the first fertilization, so that it form the embryo, so that it form the diploid embryo. N plus N, male plus female, diploid, 2N stage. The second fertilization is called second male gamete fusing with the polar nucleus, fusing with the polar nucleus in the embryo sac. The fusion with the egg cell of the first male gamete is called forming the embryo is called as syngamy. Fusion of second male gamete with the polar nucleus, that means already 2N is there, 2 nucleus are there, 1 nucleus, 3. 3 nucleus are fusing, that's why triple fusion. This triple fusion is first reported by Navasin. Double fertilization first reported by Strasburger on monotropa plant. And in this, 2 steps are there. The first step is syngamy, second step is triple fission. These two fertilizations are taking place, that's why calling it double fertilization. And the phenomena, this process where the pollens, that is reaching toward the ovule through the siphonous-like style, that process is calling as siphonogamy. This type of the fertilization, which is happening in angiosperm, is called as siphonogamy. That means pollen reaching toward the ovule. Here you see microspore, that means male gamete fusing with the female gamete, coming through the tube-like structure. This type of the fertilization is calling it as siphonogamy. What type it is? Siphonogamy. Siphonogamy type of the fertilization, that is there in the angiosperm. That first pollen tube, germination of the pollen tube, first discovered, observed by MEC. First pollen tube, germination of the pollen tube in the stigma toward the ovule. You see the pollen is pollen here by the pollination. Put pollen on the stigma. The germinating of the pollen and reaching toward the ovule. It forming the tube-like structure, that pollen tube. The pollen tube formation is taking place for the fusion of male gamete with the female. That pollen tube formation through the tube-like style is called as siphonogamy. This first observed pollen tube siphonogamy by who is that? Who is that? MEC. Observed by MEC. Okay. And then you can see the process in the fertilization. What are the steps? Process involved in the fertilization. First one is germination of pollen to form the pollen tube on stigma. Whenever the pollen reach, you see, there is a gynoecium, ovary, in the ovary, ovule are present, style, and stigma. Okay, this is the ovary. Whenever the pollen fell on the stigma, that is called as pollination. Reaching toward the stigma is pollination. That is the first step, premier step in the fertilization. And then the pollination also two types are there, self-pollination and what is that? Two types of the pollination. Pollen reaching toward the same flower stigma, pollen reaching the other flower stigma. Okay. Whatever it is, the mechanism, reaching of the pollen toward the stigma is called as pollination. And after pollination, fertilization process will start. Here, whatever the pollen which fell on the stigma, we start germination. It forms the pollen tube. The pollen tube formation is the first step. And growth of the pollen tube in the style. The pollen tube, whenever it is formed, it grows in the style toward the ovule. And the direction of pollen tube toward the micropyle end of the ovule. Andadwaram vaipuku. Reaching toward the micropyle end. Whatever the pollen tube which having the two male gametes that reaching toward the micropyle end of the ovule is the third step. And the next entry of the pollen tube into the synergy of the embryo sac, egg cell operators, toward the egg cell operators is the fourth step, important step. And discharge of the male gametes in the embryo sac. Discharge, two male gametes are there that discharge the male gamete, especially in the synergy of the first one will fuse with the egg cell, and the second one will fuse with the polar nucleus or secondary nucleus. These two male gametes fusing with the two different cells. One is with the egg cell, second one with the polar nucleus. These two time fertilization is taking place. That's why we're calling it as double fertilization, syngamy and triple fusion. This all the process is called as fertilization. All the process is called as fertilization. These fertilization steps you have to write, for that you will get the marks. Okay. There, just a second. Okay. This all the process is called as fertilization. Okay. This all the steps we will see here in this. First, pollen reaching toward the stigma, that is pollination. You see, the stigma, it facilitates the pollen to make it wet. Whenever the stigma will be in the wet condition, whenever the stigma apical part of the gynoecium pistil, if it is in wet condition, then only the pollen will be touched. Pollen will be add to this stigma. Here, there is a sexual incompatibility also is there, whether it is pollen or wet or dry, or stigma is wet or dry. These two structures decide the compatibility, whether it is compatible with the pollen which are reached the stigma or not. There is a rejection and recognition protein reaction between the pollen and stigma. Whatever the pollen which fell on the stigma, there is a reaction will take place in between these two, stigma and pollen. Whether it is recognized, okay, acceptable, same type of the genotype or not, or if not, it will be rejected. Whether it is accept or reject, that is depending upon the protein which are there on the pollen and upon the stigma. This incompatibility usually between the interspecific plant or intraspecific plant. Okay. That happen here in the fertilization at the stigma. That all the process entry, whenever the pollen deposited, that whether it is accepted or rejected, or the pollen tube formation is taking place or not, that will be depending upon pollen and pistil interaction. That means pollen and stigma interaction, whether it is accepted or rejected. Based upon that, it will be depends whether the pollen tube start germination or it dry and die the pollen. Whether it is promoted the pollen for the germination or it inhibits the germination and growth. Based upon that, sexual incompatibilities, sporophytic sexual incompatibility and gametophytic sexual incompatibility. If the incompatibility caused by this pollen, that is gametopoietic incompatibility. If incompatibility is caused by the stigma, that is sporopoietic incompatibility. That also important question. Sexual incompatibility in the plants. Two types: sporopoietic incompatibility and gametopoietic incompatibility. Incompatibility means unacceptance, not accept the pollen germination due to genetical variation or any environmental reasons or any chemical protein variation in the pollen or in the stigma. Okay. And germination of pollen grains. Whenever the pollen is accepted, the pollen start germination. That start germination. Okay. Whenever the pollen start the hydration, water, wet, that entire content that are moved to the pollen tube, that start movement, start growing of the pollen. You know, one important question in a competitive exam they ask, the pollen tube germination is depending upon which element? Boron. And the boron and sugars, these two content is determine or initiate the germination of the pollen tube. The boron content is less, the pollen germination is stopped. Okay. Sometimes there is a cut of pollen tube vacuole. It stopped with the callose plugs. It stopped with the callose plugs to stop flowing of the gametes toward the tip. That callose blocks, we call it as cap blocks. These are blocking to stop the transport of the microspore toward the pollen tube end. Okay, so that it will be cause for the sexual incompatibility. Here you can see the pollen structure also you have studied now. Pollen structure, the exine and intine, it having exine outer layer, it is made up with sporopollenin. Intine is made up with pectocellulose. It having the nucleus and ornaments. The sporopollenin is a very hard, tough cell wall which is there upon the pollen. That's why pollen can survive even long time without any destruction, even in the rain, even in the water, even in the dry, even in the high light. Also, it can't destruct because that hard coated sporopollen is present on the pollen. Okay, you see the pollen. What happened here is the pollen microspore mother side divided into two. That is, what are those? What are those two? Generative cell and tube cell. You see the generative cell again dividing into two gamete, two male gamete. The two and the other one is the tube cell. The tube nucleus and two male gametes. These three nuclear stage, the pollen choosing with the pollen germination which takes place. In the pollen tube, three nucleus are there, that is pollen nucleus and two male gametes. Pollen nucleus, it initiates the pollen tube, pollen germination, and it forms the pollen tube, and the two male gametes fuse with the one with the egg cell, one with the secondary nucleus. This three nuclear stage is there in the germinated pollen. Okay, that also very important. Which nuclear stage? Three nuclear stage. Usually in the gymnosperms only in the two nuclear stage it releases. But in the angiosperm it releases in the three nuclear stage. Okay, that you should remember. The three nuclear stage. Okay, and then you can see the entry of pollen tube in the style. Whenever it started germination, whenever it is recognized, whenever it is compatible, whenever it is not incompatible with the stigma, whenever it is compatible, whenever it is recognized, accepted by the stigma, start germination, and the germination pollen tube with the three nucleus, it grow in the style. That is in the tube-like structure of the gynosium. It grows in the style. Okay. Usually there is a three type of the styles which are hollow or open type of the style and solid style and semi-solid or half closed style. Okay, hollow, fully hollow, open type, this one. Or solid type filled with the other tissues. Or semi-solid of fill or liquidization type. Sometime I already told you Carlos plugs are formed cap blocks so that the pollen tube not reach toward the excel. That also one of the incompatibility I told you. Sexual incompatibility. That also one of the sexual incompatibility I told you. Okay. But also one of the most important, I already told you, in the compatibility, in the recognition. And then you see the second step also completed. And then the next, you see here, sometime what happened in the pollen tube? Whenever the pollen germinated, in the pollen tube, there is a, what happened? There is a callus, cellulose plugs, which are blocked in the pollen tube to stop the male gametes toward the stigma, or toward the ovule. That callus flux formation also one of the sexual incompatibility process. You see here, whenever the pollen is start germinated, what happened? You see the Golgi bodies, endoplasmic reticulum, vesicles, lipid, particles, and starch granules, storage food material. These all grown. Where the part, the exine, not the exine, intine is forming the pollen tube. Exine here is fit by the enzymes and the intine is forming the pollen tube. That means intine is made up of it, pectocellulose. The pectocellulose made of tube is forming that sometimes it blocks the tube that is callus plugs. And then the next entry of pollen tube into the ovule. Into the ovule. Still now the pollen tube reached toward grown in the style and then it reached the ovary. From the ovary, in the ovary, it reaches toward the ovules. How it is reaching? Yeah, based upon the way process of the reaching of the pollen toward the ovule, there is a three types that are porogamy, chalazogamy, and mesogamy. Okay, porogamy, chalazogamy, and mesogamy. The porogamy is the commonmost type. Usually, the pollen which reach to the ovule through the micropylar end, andadvaramu. opening space of the ovule, that is micropyle. Micropyle end, it is the commonmost. From the open porous space, the pollen reach toward the ovule. Open porous space, the micropyle. That open porous space of the integument is called as micropyle that is reaching through the four randrum. Four. That's why they call it as porogamy. It is the commonmost type of the fertilization type. You see, the next is chelagogamy. The pollen tube reaching toward the chelaja, not from the micropylar end. This is the micropylar end. This is the chelagal end. Where the integuments having the porous space, that is micropylar end. Where the integuments is open up, that upper area is called as chelagal phase. That means here what happens? The pollen which are reaching whatever the pollen which are reaching toward the ovule through the chelagal end, not from the micropylar end. Now, if it is end, if it is reached through the micropylar end, that is porogamy. Not reaching, if it is reached through the pollen tube, if it is reached through the chelaja part, that we call it chelagogamy. This chelagogamy was identified in the Casuarina and an Amantipare family. Okay, this is the second type of the fertilization, and then reaching of the pollen toward the ovule. The third type is mesogamy. Pollen tube is reaching through the funicle or through the integument. Integumental layer, reaching toward the integument layer toward the ovule. That is calling it as mesogamy. Okay. Though it is coming from the funicle or from the integument, it must reach through the micropylar end only. Okay. Whenever, whatever the way, the pollen reaching toward the ovule, but it must reach the ovule through the micropylar end only. That is a common most type porogamy. Okay. Here you can see the types. Three types: porogamy, chelagogamy, and mesogamy. Mesogamy is usually happen in the all Cucurbidaceae family, is the common most type. And above all the three types are there in Brassica oleracea. Three types of the reaching of the pollen toward the ovule are present in Brassica. Avalu, Brassica nigrum, rape seeds. Brassica. Three types are present in the Brassica. Okay. And then you see reaching of the pollen toward the ovule sometime may not be happen at that time. Some of the extra ovular structures. Yesterday we have discussed in the ovule structure, obturator, epistage, hypostage, piliform apparatus. This all we have studied in the ovule structure. That special structure also will be useful to reach the pollen tube into the ovule, to entry of the pollen tube into the ovule by forming a short bridge. These are finger-like projections. These finger-like projections which are forming at the micropylar end. You see, the obturator which is forming the finger-like projections at the micropylar end. So that two sides of the micropyle, it having the finger-like projection. This finger-like projection, it carries the pollen toward the synergies. toward the synergids. That is called as obturators. After fertilization, this obturator will be shrink or disappear. Only for the fertilization only it will be useful. Okay. And then the next is filiform apparatus, also one of the supporter. These are finger-like projections. You can see the finger-like projections which are there in the synergids. You see in the Excel apparatus, three cells are there now. First one, the middle one, big one is the Excel operator, Excel, and the two sides, two synergids are there. Three cells, three nuclei. But the pollen tube it not reach directly toward the Excel. First pollen tube, it reach toward the reach toward the ovule Excel through the synergid cell, through the synergids. From this synergid, in the synergid, filiform operators, finger-like projections are there that absorb this male gamete and it send, it transport, it give the way, it give the way to the male gamete toward the egg cell. That here it is helping to reach the egg cell, male gamete reach toward the egg cell. These are also supporting tissues. These are the transportation material to observe, to transport the male gamete toward the egg cell. These two ovule structures, that is obturators and filiform operators. Obturators are the micropyle outside of the ovule, and filiform operators inside the ovule in the synergids. Okay, these two are helping in the fertilization. You see, another one, the X bodies. You see, after discharge of the pollen tube into the embryo sac, sometimes the what happened here, the X bodies also appearing along with the male gametes. Along with the male gametes, the X bodies also presenting. That X bodies nothing but the vegetative decomposition products. You see, these also, their X bodies are sterile bodies or vegetative bodies which are present in the ovule embryo sac. You see, the next step, entry of pollen tube into the embryo sac, into the embryo sac. Till now we are studying up reaching the embryo sac, ovule only. The ovule, whenever it reaches towards ovule, and whenever it reaches the synergids, companion cell, synergid cell, the pollen tube, whenever it reaches the synergids, and there it discharges the two male gametes in the synergid. It discharges the two male gametes in the synergid. The two male gametes of that, one male gamete fuse with the egg cell, and other second male gamete fuse with the secondary nucleus. That is basically depending upon chemicals which are present. Chemotropic substances. Chemotropic chemicals, same compatible chemicals present or not. Based upon that, whether it is compatible or not. And obturators also help, and filiform operators also help to reach the pollen tube toward the egg cell apparatus. Okay, so the pollen tube growth will take place up to the synergid cells only. It can't grow behind the synergid cells. There only it stops. It discharges the pollen male gametes in the embryos. Okay. There, whatever the two male gametes fission takes place two times. That's why we call it as double fertilization. Double fertilization. Two different fertilizations. You see, I already told you the fusion of male gamete with the egg cell, that is syngamy. That process is called as syngamy, so that it forms the zygote embryo. It forms the zygote embryo. That process is called as syngamy. This process first observed by Strasburger. This process first identified by Strasburger and Fortloka plant. And the second one is the Lilium fritillaria male gamete you see fusing with the secondary nucleus or polar nucleus, which are there in the central cell. The polar nuclei fuse with the secondary male gamete, second male gamete, so that it forms the 3N structure, three nuclear structure, triplex structure that we call as primary endosperm nucleus pen, not writing pen. Primary endosperm nucleus, which form the endosperm. Endosperm is a nutritive tissue which will be useful to germinate, to develop, to mature, to germinate the seed. That's endosperm. Endosperm will be formed after fertilization only with the fusion of second male gamete with the polar nucleus. That process is called as triple fission. Triple fission. These two fertilization, one is syngamy, second one is triple fission. These two processes commonly called as double fertilization. These two processes commonly called as double fertilization. Okay. So that here after fertilization, embryo is forming, and endosperm also forming. Diploid embryo, triploid endosperm. Endosperm not in always in all the plants triploid. Suppose in the drusa, it is in 9x. Suppose in the fritillaria plumbagella, it is 6x. Like that. There is a variation is that. 5x, 5n, 6n, 9n. Endosperm is present based upon the polar nuclear stage, whether single polar nuclei or two polar nuclei or four polar nuclei or nine polar nuclei. Based upon that, 8 polar nuclei. There is a various type stages of the endosperms are there. Usually triploid. Usually triploid endosperm because triple fission, three nuclear fission. This endosperm will be useful in the maturation, developing of the embryo. But whereas in the gymnosperm, it is applied. Endosperm is applied. Why? Because it is forming before the fertilization only. Before the fertilization only, it is forming the endosperm. That's why before the fertilization, from the some of the female gametophyte cells are forming the endosperm. That's why gymnosperm endosperm is haploid, angiosperm endosperm is triploid. In the so many competitive exams, we ask the question. Usually, angiosperm endosperm is haploid, diploid, triploid, tetraploid. Triploid, three nuclear stage. Usually, gymnosperm endosperm is haploid, diploid, triploid, tetraploid. Haploid. Why? Why the gymnosperm endosperm is haploid? Why the angiosperm endosperm is diploid? Because in the gymnosperm, what happening? In the gymnosperm, before the fertilization only, female gametophyte is forming the haploid female gametophytic cell is forming the endosperm. That's why it is haploid, one and in stage. Whereas in the angiosperm, the two nucleus, polar nucleus, and one male gamete. Three nucleus are fusing, that's why triploid. Okay, and sometimes the polyspermy also happening. That means more than two male gametes. If the more than two male gametes are releasing through the pollen tube into the ovule embryo sac, that is polyspermy. Sometimes not the single pollen tube, sometimes more pollen tubes may be formed. Not a single, two, three, four pollen tubes may be formed. Usually, whenever the one pollen tube starts germinated, the remaining pollen tube germination will be stopped by the callus slug there only at the stigma only. But sometimes the more than more pollen tube formation also will take place. At that time, some first pollen tube male gamete may fuse with the egg cell that form the embryo, but second pollen tube may be fused with the synergids or with the antipodal cells. May be fused with the synergids or fused with the antipodal cells other than the egg cell. That time it leads to the polyembryony. Usually single embryo is formed by fusing of the male gamete with the egg cell. Sometimes extra pollen tube may fuse with the polar nuclei. It's called heterofertilization. Extra pollen tube sometimes fuse with the polar nuclei, secondary nuclei. That called as heterofertilization, not homofertilization. These are the types happen by three ways. What are these ways? If the more pollens are released, if the more gametes are released in the embryo sac, or if the more pollen tube germinated enter into the embryo sac, or pollen tube may fuse with the sinner secondary nucleus, polar nucleus. Okay, these three ways there is a chance of polyembryony. You see here what is happening? Fertilization process, all the double fertilization process here is the egg cell, the synergid, okay, and here antipodal cells at the cellulose end, egg cell apparatus at the micropylar end. One egg cell, two synergid. Okay, here the pollen germinated, it formed the pollen tube, it reaching towards the ovule through the micropylar end, porogamy usually. You see the releasing of the sperms, male gamete, two male gamete toward the embryo sac. You see what happening here? Here, one male gamete fusing with the egg cell. One male gamete fusing with the egg cell. It is forming the zygote. Second male gamete, you see the second male gamete going toward the polar nucleus. The two polar nucleus plus second male gamete is forming the endosperm, triploid endosperm, triploid endosperm. The two petalization, this is the one, this is the two. Two petalization. The first petalization we calling it as syngamy, so that it is forming the zygote and it form embryo and seed. And the second fertilization, male gamete with the secondary nucleus, that is we calling it as triple fission. With that it is forming the primary endosperm nucleus stem. It is forming the endosperm. There is a nucleus structure for the development mature of the embryo. And syngamy plus triple fission both commonly called as double fertilization. Two-time fertilization is taking place. The fertilization is taking place in two times. That's why we're calling it as double fertilization. This happens only in the angiosperm, not in the gymnosperm. The gymnosperm is only one fertilization, fission of the male gamete with the female only. Okay, only in the two nuclear stage, pollen tube will be germinate, pollen will germinate in the gymnosperm. But whereas in the three nuclear stage, it germinates in the angiosperm. One is the two nucleus, two male gametes. Okay, fertilization has taken place. After fertilization, what the process? The fertilization, what happened? The zygote form. The zygote will become the embryo. This embryo formation will take place. The embryo. In embryo, what happened? It divides into three stages of the three layers, that is protoderm, ground meristem, and procambium. Each one, the protoderm will be formed the outer layer, and the procambium, ground meristem, which form the meristematic tissues, and the procambium, which form the cambium at the middle. Okay, you see the where it is the embryo, seed, the embryo, dicot embryo. You see the cotyledons and root apex and shoot apex and the suspension. You see the endosperm, which are using for the maturation development of seed. You see what are the steps, what are the modifications are happening after fertilization? Endosperm, embryo development, formation of seed and fruits will take place. This all process is called as post-fertilization stages. You see, after fertilization, sepals, petals, style, stigma, all will be fell down, all will be dried or detached. And ovary will become the fruit. Ovule will become the seed. Ovule will become the seed. Egg cell. Egg will become the zygote after syngamy. Egg will become zygote. Ovule will become the seed. And all the ovary will become the fruit. You see, this is the ovary. This is the ovule. This is the egg cell. Egg cell will become the seed. Ovule will be, egg cell will become the zygote, embryo. And ovule will become the seed. Ovary will become the fruit. This is the processes will take. Tunicule will become the stalk of the fruit, and micropyle will become the micropyle of the seed for the facilitation of oxygen and water uptake. And nucellus will be utilized for the maturation of the seed. If the remaining nucellus is there at the middle, this one nucellus, the remaining nucellus is there. The remaining nucellus is called as perisperm. The complete exam are asked. Perisperm is the nucellar part or endosperm part or gametophytic part. Nucellus. Okay. Nucellus types also is there. Crossing nucellus, tendon nucellus, we have studied. Okay. Endosperm types also is there that we will discuss. Cytoplasmic, nuclear, ruminate, uh, helobial type of the endosperms. Outer integument of the ovule will become the testa. That means outer seed coat. Ovule, by usually ovule types also we studied. Ategmic, unitegmic, bitegmic. Ategmic are mostly present in the Ranunculus, Plarici, in some of the gymnosperms. And the bitegmic is common in all the angiosperms. But outer layer will become the testa, and inner integument in the ovule two layers. Outer layer, outer integument will become the testa, and inner integument will become the tegmen in the micropylar space, open porous space. Here it is the ovule will become the total seed. Here what is the embryo? It is there inside. Okay, and synergy cells will be degenerated. Secondary nucleus we form the endosperm by the triple fission, and antipodal cells will be degenerated. These are only vegetative cells, no use in the reproduction. Sometimes maybe the second male gamete may fuse with the antipodal cells. That time it may cause for the polyembryonic. But usually endosperm, antipodal cells are vegetative cells only. It not participate in the fertilization. Here you can see how it is taking place. You see from the anther, the from the andosium, anther. You see anther GS also you have studied anther LS. The sporangium, microsporangium, microspores are releasing after meiosis from the microspore mother cell MMC. The microspore which are forming the pollen, the microspore that having the three nuclear states, one tube cell and two male gametes that is reaching toward the stigma, pollination, and the pollen germination, and the pollen tube formation, and the next is reaching toward the micropyle, reaching toward the ovule, reaching toward the embryo sac, and double fertilization. These are the steps. You see the ovule, which are there in the gynosia, ovary. The ovary ovule is present. In the ovule embryo sac, you can see the 8 nuclear states, egg cell apparatus and antipodal cells and polar nucleus. This egg cell will fuse with the male gamete, first male gamete, and the second male gamete fuse with the polar nuclei is from the endosperm, and the zygote will become the embryo, and ovule will become the seed. Ovule will become the seed. The zygote will become the embryo. This is the overall fertilization process which is happening in the angiosperm, which is happening in the angiosperm fertilization. That is happening. That is the fertilization types, fertilization process. Okay, and sexual incompatibility also we have discussed. Sporophytic sexual incompatibility and gametophytic sexual incompatibility. Okay, that also important question. Fertilization process also important question that we have studied. Okay, and then we will discuss the next. We want to no. Okay, these are the steps. What are those? Germination of the pollen tube to form the toward the stigma, growth of the pollen tube into the style, direction of the pollen tube toward the micropyle, and entry of the pollen tube into the one of the synergy cells, and discharge of the male gametes, double fertilization. These 6 steps are there in the fertilization. But sometimes the fertilization without the fusion of the male and female gamete also will be happen. That we calling it as apomixis. And the without the fertilization, the fruit also will be formed sometimes. That we call it as parthenocarpy. Apomixis and parthenocarpy. Without the fertilization, the fruit is formed, that is parthenocarpy. Parthenocarpy first identified by whom? Null, 1902. Usually parthenocarpic fruits are seedless fruits. There is no seed. Why? Because there is no fertilization is taken place. There is no fusion of male and female. There is no syngamy. There is no zygote formation, no embryo formation. So there is no seed formation. Nowadays seedless fruits market is more. Seedless fruits, banana, seedless tomato, seedless lemon, watermelon, grapes, cucumber, the seedless fruits which are forming by the parthenocarpy. Without the fertilization also, fruits formation is taking place. It is resemble the normal production of the fruit, but seedless. Only one thing is seedless. Many commercial fruits are made seedless now. Banana, grapes, watermelon, like some of the fruits we are preparing by using this parthenocarpy technique. Instead of normal fertilization, the normal fertilization seeds are formed in the fruit because of fertilization. But without the fertilization also, fruit formation is happening. That process is called as parthenocarpy. Okay. In the parthenocarpy, what is happening in the parthenocarpy? You can see there is a two types, vegetative parthenocarpy and stimulative parthenocarpy, stenospermocarp. Two types are happening. Sometimes pollination stimulation is needed for the beginning of the fruit development. That is called as stimulative parthenocarpy. If there is no need of any pollination, if there is no need of any pollination, that is calling it as, that type we are calling it as, what it is? Vegetative parthenocarpy. Vegetative parthenocarpy. That is common in cucumber, banana, watermelon. Okay. Sometimes, though fertilization is not needed, some amount of the pollination stimulus needed. That means pollen reaching toward the stigma. Though it is not germinated, though it is not fused with the egg cell, but pollination stimulation is needed to form the stimulative parthenocarpy. That is called as stenoparthenocarpy. That is common in watermelons. Okay, these are the two type of the parthenocarpy. Niche classified the parthenocarpy into two types, that are genetic parthenocarpy and environmental parthenocarpy, chemical induced parthenocarpy. Okay, what are those types? Genetic parthenocarpy, that means due to hybridization or mutation, the parthenocarpy may be happen. That is common in citrus fruits, cucumber, cucurbita, gomadido, sabira, sora, kakara. The cucumber and in the citrus. This is due to hybridization or mutation. genetical variation. It may be happen the parthenocarpy. Second one is environmental parthenocarpy. Environmental condition may be frost, cold, fog, low temperature or highest temperature. It leads to the parthenocarpy. If the low temperature up to 19 hours, it induces the parthenocarpy. In the fierce fruits. If we grow in the low temperature areas, it leads to the parthenocarpy. No need of the fertilization. And the next one is chemically induced parthenocarpy. Apply of the phytohormone, gibberellin and auxins. Gibberellin and auxins. This induced the chemically induced parthenocarpy in the grapes by spraying the auxins and gibberellin, IBA and gibberellic acid. You know, gibberellin from Gibberella fusicoride, fungi, Buccaneer disease. That chemical gibberellin, it induces the parthenocarpy, it induces the more bunch of the grape, it induces the fruit, improve the fruit size and bunch size of the grapes. Okay, it also induces the germination of the seeds, that is gibberellin. So these are the three types: genetic parthenocarpy, environmental parthenocarpy, and chemically induced parthenocarpy. You see, parthenocarpy induced by so many ways that I told you auxins by using IBA. Usually, the parthenocarpy, sometimes it's needed the artificial pollination. That means no need for the pollination stimulation, no need of the germination. That means with the dead or altered pollens, but just for the pollination stimulation. Or pollinating the stigma with foreign pollen grains. It can't germinate. Foreign pollen grains can't germinate on the stigma of flower. Same genus, same species pollen only will germinate. But it stimulates stigmas, extract pollen, or some other molecules. Okay, and synthetic growth of substance of paste forms, injection, spraying. These all are different mechanism for the parthenocarpy, stimulation of the parthenocarpy. You see the diploid banana from Asian parents. Seedless from Musa accumulata and Musa bulbosiana. There is a Musa paradisiaca hybrid banana, what we are eating now. Seedless banana. Applied set of chromosome of Musa accumulata and another applied set of chromosome from Musa bulbosiana. So that we are forming what we are eating now, banana, seedless banana, that is hybrid banana, that is caused by producing by through the parthenocarpy. There is a lot of significance, importance is there. Seedless fruits now having the great significance, good source. So many are preparing seedless fruits. In horticulture, the seedless fruit formation is very good, extraordinary source to produce more fruits. Seedless fruits usually having the great commercial importance than the seed fruits. These are seedless fruits are useful in the preparation of jam, jelly, sauce, juice, fruit drinks by using these seedless fruits. High proportion of edible part is there in the seedless fruits. So there is no seed, so all the part will be edible. Increase production even in the adverse condition. Even in the adverse condition also it increase the production. Why? There is no need of the fertilization. Even in adverse environmental condition, low temperature or high temperature or pre-chilling or in heavy fog or heavy smoke, time also this parthenocarpy will be happen. There is no loss of fruit formation. And protected and reduced cost of cultivation. It will be protected and reduce the cost than the seeded fruits. So this is very much useful in the horticulture commercialization for product. You see, the commercial use seedless cucumbers, seedless banana, pineapple, banana, orange, grapefruits, seedless. You see cucumber, seedless cucumbers, summer squash that we eat in the summer season. You see the seedless brinjal, seedless cucumber, seedless banana, seedless pineapple. The fruit crops sometimes may be difficult to pollinate or fertilize. That time, the parthenocarpy will be useful. Usually in the dioecious species, the parthenocarpy increase the fruit production because it already no, male and female is separate now, dioecious species. So the staminate trees do not need to plant. No need of the plant of the male plant nearby. Only from the female plant we can produce the fruits. What are the influencing factors that causing the parthenocarpy? Environmental factors. What are those? I or low temperature. I or low temperature and growth regulator. Sometimes self-incompatibility. I told you sporophytic self-incompatibility or gametophytic incompatibility. Either stigma may cause for the infertility or maybe pollen cause for the infertility. Chromosomal aberration, sometimes genetical disorders, and genetic factors like genome control, mutations, hybridization, sometimes absence of pollination and fertilization, that all influence the parthenocarpy. Male sterility, degradation of pollen, mother cell, or degradation of fertilized ovules, or sometimes embryo abortion, or chromosomal irregularities during the meiosis, that all leads to the triploidy. The triploidy usually is a parthenocarp seedless. You know, if the diploids 2n, 2n or 4n only having the seeds, the triploids, hexaploids are seedless. Why? There is no fair set of the chromosomes. Uneven set of the pair set of the chromosomes are there. That's why there is no chance for the formation of the seed. That's why seedless fruits. You see, cause induction of the parthenocarpy in the fruit crop. We are preparing the triploid fruits so that there is no need of the seed formation, ovule, failures of the fertilization. And you see in the grape due to embryo abortion, in the citrus due to self-incompatibility, or cytoplasmic male sterility, or chromosomal irregularities causing. And in apple, lack of pollination, very uh, rare. The apple, the banana due to triploidy, not the diploid, triploidy. That's why it's losing the seed formation capacity. You know the apomixis without mixing of the male and female gamete, the fruits will be formed. That is apomixis. And your parthenocarpy means it also same without the fertilization. Apomixis and parthenocarpy is the same. Those are the two no fertilization. Both produce offspring similar to parents, apomixis and parthenocarpy. The both cases there is no chance of diversity of genes. Why? From the same plant, from the mother plant only, it is producing the fruits. There is no male plant, other male plant. There is no pollens. There is no male and female fusion. So only mother genes will be remain for the generations together. Both the type of phenomena do not involve in the fertilization. Both are used asexual means or vegetative propagation methods, not the sexual process today. They still use the sexual organs of the plant or ovary or ovules will be used, but it is not taking the fertilization. It's not taking the fusion, but the fruit formation is taking place. What are the difference between the apomixis and parthenocarpy? You see, apomixis and parthenocarpy both are asexual methods, but need of sexual organ, ovule. Apomixis is the formation of seed, whereas the parthenocarpy is the formation of fruit without fertilization. Parthenocarpy. Carpy means fruit. Formation of the fruit without the fusion, without the fertilization. Okay, apomixis, without the fusion, seed formation, embryo formation. Apomixis produce genetically identical mother cells. Parthenocarpy also produce genetically identical upsprings, next generations. Apomixis is seen in angiosperm and gymnosperm, and parthenocarpy is seen in plant and animals also. Parthenocarpy in animals called parthenogenesis. Okay. Parthenogenesis means whenever the egg cell is not fertile, unfertile, that unfertile egg cell grown, develop as an embryo. Unfertile egg cell developing as an embryo. That is parthenogenesis. Applied parthenogenesis. That means only applied female gamete or applied male gamete develop into an embryo. Diploid parthenocarpy means in the many plant megaspore mother cell does not undergo the meiotic division. So that is diploid only, still diploid. Does not taken the reduction division. So diploid nucleus only is present. The diploid nucleus will become the diploid embryo. So that egg always diploid. The diploid unfertile egg it form the diploid embryo. Okay, these are the causes for the parthenogenesis. What are the induced parthenogenesis? Parthenogenesis is induced by creating low temperature or high temperature or exposing with X-rays or injecting certain chemicals. With that, we can induce the parthenogenesis. What is the difference between parthenocarpy and parthenogenesis? Development of fruit without fertilization. Development of new organism without fertilization. Okay, seedless fruits are produced. Your organism are produced. Female clones cannot reproduce sexually. Offsprings are not produced in the parthenocarpy. The haploid offsprings form in the. Why offsprings are not formed in the parthenocarpy? Why not? Why? Because seedless, no seeds. It occurs only in the plant. It occurs in animals, plants. These are there in the banana, watermelon, pineapple, cucumber. These are there in the lower plants, insects, and lizards. You see the difference between the normal fertilization, parthenocarpy, and parthenogenesis, apomixis. You see, the floral development will take place in the parthenocarpy, fertilization, apomixis. But pollination and fertilization will take place in the normal fertilization. Here, the fertilization will be stopped, ovule development will be arrested. Here, in the pollination, fertilization will be take place. Here, ovule development are arrested. Even in the apomixis and parthenocarpy, the both. The both is happening. Why? Without the fertilization, without the fusion of the male and female happening here. Post pollination, floral development be there. The floral development is not there. Here you can see the fruits formation how it is. Fruit development without the seed is parthenocarpy here. Why? Because without the fertilization, without the embryo formation, autonomous fruit and seed development will take place in the apomixis. Though it is not mixed, the only female mother cell, mother plant ovule will become the fruit. Okay, this is the seedless fruit and seeded fruits. You see, seedless and seeded fruit both are there in a market. Seedless fruit is the parthenocarpy. Seeded fruit is in normal fertilization. Okay, this is happening in our side. Okay, thank you. With this, I'm concluding. Okay, next we will discuss in the next class uh, polyembryony that we will discuss, and endosperm, polyembryony and endosperm, and embryology in relation to taxonomy, horticulture, and applications of palynology. These four chapters we will discuss in the next week. Okay. Okay. Thank you. Today we have discussed fertilization, apomixis, parthenocarpy. These three essay questions very important. Apomixis, parthenocarpy, huh? Very important question. These are very important. You go through this and you can easily know this all the steps. Okay. Apomixis, apo without mixing. Aplospory, diplospory, and adventitive embryony. Okay. These are the two types, recurring and non-recurring. Recurring is mainly vegetative propagation and agamosperm. Okay. Okay. These are we have studied. Diplospory directly from the megaspore cell without the reduction division. Okay. These are types are there. Taraxacum, ixarum, antennaria, allium types are there. You see, in the prophase, it arrests the division so that it forms. In the metaphase type, at the latest time, in the 8th phase nucleus, the embryo, it directly forming the embryo. The attenuated after a long interphase acts as a functional megaspore. Apospory, it is forming by somatic cell. If the megaspore mother cell undergoes normal meiosis, at that time, gradually the pore maybe disappear. That time, the nuclear cell, nuclear cell or embryo sac, other cell will cause for the germination. Instead of egg cell, embryonic cell, other cells, either chelagel cells or antipodal cells or nucellas that forming the embryo. Okay. Adventive embryonic, other than the embryo cells, embryonic cell, other than the ovule, the remaining part it is taking in the formation of the fruit, that is adventive embryos. You see, that is common in adventive embryos, is common in citrus, mango, Mangifera indica, and citrus. Okay, these all are the types we have studied: applied parthenocarpy and diploid parthenocarpy. You see, apogamy, parthenogenesis, pseudogamy, and semigamy. These all we have discussed. Okay, apomixis, parthenocarpy, fertilization. These all are very important topics which are coming in the examination essay questions. Okay. Okay then. Bye. Okay, right.